Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapelli, Assistant Professor at Department of Pharmaceutical Science and Technology, Institute of Chemical Technology, Mumbai. So, in our previous lectures in nanoparticle as a novel drug delivery systems, so we have discussed various inorganic nanoparticles. In that we have discussed the introduction on inorganic based nanoparticles, and we have seen the advantages and the importance of the inorganic based nanoparticles. And after that we have discussed the unique properties of the inorganic based nanoparticles. Specifically, we have discussed their optical properties, and also we have discussed electrical properties, and also we have discussed the magnetic properties of the inorganic nanoparticles. In that we have discussed three examples: the gold, silver and iron oxide nanoparticles. And at the end we have seen the FDA approved nanoparticles in the previous slides. So, today we are going to see the evaluation of the nanocarrier systems. So far we have discussed various nanosystems. We can see from the polymeric nanosystems to the lipid based, and in the lipid we have seen liposomes, SLN and the nanostructure lipid carriers. And after that we have seen various vesicular systems like neosomes, ethosomes, transferosomes and the bilosomes, and we have seen various types of the vesicular systems. And after that we have seen the inorganic nanoparticles also. So far we are focusing on the properties and the preparation method, and then we have seen the application and importance in the drug delivery as well as in the imaging applications. So, with that which will the nanocarrier system or nanoparticle as a drug delivery system will not end. After the preparation and before doing the application, we must characterize our material. So, today we are going to focus on what are the characterization studies of the nanosystems. So, these are all evaluation techniques, or you can say the characterization methods which can applicable for the all type of nanosystems. So, these are the general evaluation studies. So, today we are going to discuss on the various types of the evaluation properties of the nanocarrier systems. So, first the evaluation study, that is physicochemical characterization, and then we will see the in vitro studies, and after that what are the in vivo studies required for a nanocarrier systems, and then we will focus on the what are the clinical studies, what data need to be provided before doing the clinical studies, that we will see. So, in the pictorial representation you will see the various steps involved. Step 1, and it indicates the chemical and physical characterization. Now, you can see here we are going to see what is the composition, what is the degradation time, and what is their structure, and we can see the shape, size, what is the roughness of the material that can be detected by using the various microscopic techniques. Okay. So, here you can see the picture that speaks about the physicochemical characterization, or you can say the morphology of the material. Now, once it is done, we will go to the second step, that is in vitro test. So, in vitro test, typically here we will see the drug release, and we will see the stability studies, and also we can see some of the studies which we can perform, like activity studies. Here is the some of the examples: cytotoxicity, the genotoxicity, and if you want to prove some activity studies, for example, if you want to do anti-accident study, and if you want to prove anti-cancer studies, and if you want to prove your material is showing as a antibacterial. So, such studies can perform in the in vitro test. Now, we know that in vitro these are all are we will perform in the artificial containers, or you can say the in the glass containers. Okay. We take the enzymes, we will take the proteins, and we will take the cells, and we do the interaction, and then we will see the outcome. So, yes, we will mimic the physiological conditions, but this can be performed in the glass containers. Whereas in vivo is nothing but these studies performed in the animals. It may be rat, the pigs, even in the monkeys, rabbits also. So, here we try to understand the efficacy, safety of the material in the in vivo test. We can also perform pharmacokinetic studies also. So, these are the few examples: sensitization, irritation, systemic toxicity, and the implantation. And once you done this up to this, then we can go further for the clinical trials in the humans. From that, what we will get? So, we will get the safety efficacy data on the human beings. So, but if you are going to the clinical trials, you must provide this preclinical data. So, then only the regulatory people will allow for the approval of the clinical trials. Yes, this can be performed in the huge number of the patients and healthy voluntaries, and at different ages, and different gender, and different ethnicity. So, with that you will understand the effect and safety of your new drug, and as well as nano formulations. With this, we will see today's session. So, the first one is the physicochemical characterization. So, yes, we are claiming that our formulation is in the nano size, right. So, when you are claiming in the nano, you must see the size of the particle. So, particle size distribution, the dynamic light scattering DLS technique can be used, which will screen your formulations. This technique used for the screening of the various formulations, and if you want to optimize, so DLS is the one of the best technique. The principle it involves, it measures the random or Brownian motion of the particle in the liquid, so which comes in the result, that is a hydrodynamic diameter of the particle. So, this is the instrument which looks like a UV cabinet, UV spectrophotometer. So, here you need to put your sample in the cuvette. So, now here you can see, right. So, once you after the keeping of the sample into that, so now you can see the laser source comes, and that passes through the attenuator, and then the light will pass through your sample. So, it measure the scattered light, so that is through the detector, that will give the results in the nanometer. Now, we can see the result Z average diameter in the nanometer. In this picture, you can see the particle size is 63 nanometer. So, that is the indication of the hydrodynamic diameter of the particles, nothing but it will not give the particle size of single particle as you get in SEM, as you get in a TEM, and as you get in a AFM. So, here it will not give the particles of single particle, we need to disperse your material, your nanoparticle in the cuvette, and then that cuvette need to be placed, that will give the average hydrodynamic diameter of the particles, which is suspended in your liquid medium. So, that is the limitation of the DLS instrument. Okay. So, further we can also measure the polydispersity index. So, here the polydispersity index indicates the width of the particle size distribution with lower PDI values, suggesting a more monodispersed, narrowly dispersed sample. So, here if the number is very less, the homogeneity is the very high. So, the polydispersity index always near to 0, that speaks about the distribution of your particles. So, with the same instrument and during the particles analysis only, you will get the results. If your formulation, the suspension is particle distribution is very less, and you will get the lower PDI value. Now, the third analysis, zeta potential analysis. So, with the same dynamic light scattering, you can also measure the zeta potential, but you need to change the cuvette that will measure the electric potential of your formulation. So, what is the meaning of zeta potential? Zeta potential, also known as electrokinetic potential, is a measure of the electrical potential at the boundary between the charged surface of the particle and the surrounding fluid. Now, in the picture you can see here, so this particle is the negatively charged particle. So, consider it is in a PLGA, polylactic glycolic acid, or some other carrier which comes in a negative charge. Now, you can see on the surface the positive charge is overloaded on the surface of the negative charge particle, and top of that you will see there is a boundary layer, right. So, which is represent the negative charge and the positive charge. So, that is a boundary layer. So, now what is the zeta potential is nothing but, as definition suggests, that it is the measure of the electrical potential at the boundary between the charged surface and the surrounding fluid. This is surrounding fluid. So, you can see here, so this is the indicative of the zeta potential. So, that is represented in the millivolts. Now, you can see in the picture. So, here you can see the zeta potential, the millivolt, that is in minus 11.6, right, and the results is good. Now, you can see the peak, the distribution is very good. And now, so why it is important to measure for the nanoparticles? Because the zeta potential is a key indicator of the stability of any type of the colloidal dispersions and the suspensions and the emulsion. To see the stability, your formulation should have the highest number of the zeta potential. If that is maintained for longer days, so that means your formulation is stable. So, now we have another few techniques which will measure the size and shape of the nanoparticle. With the DLS, you cannot measure the shape of the nanoparticle. Yes, you can measure the size, but you cannot measure the single particle size, that will give the hydrodynamic diameter. If you want to know that the size of each particle from your formulation, so the preferable technique is SEM or TM and the AFM. So, now you can see, we can measure the size as well as you can see the shape of the particles. You can see all these three are, the shapes are spherical, there is no non-spherical materials here, right. So, first one we are going to see the scanning electron microscopy. Now, the term itself speaks that the electrons will scan the surface of the material, and that backscattered light can be converted into the image. So, now we can see the scanning electron microscopy provides information on the sample surface and its composition, the roughness and the smoothness of the material. Now, you can see here you can see, so that will give the roughness and the smoothness of the material, and you can see it can give the particle size. So, here is the scale, with that we can consider what is the size of your particle, and the resolution is limited to 0.5 nanometer, and the magnification is very low compared with the other system, the TEM and AFM, and it is suitable for only large particle, it is not suitable for the small particles. And you can see here the noise is very high in these formulations by using the SEM, whereas TM it has a higher advanced compared with the scanning electron microscopy. So, here it will use the transmitted electrons, nothing but the electrons will transmit through the sample, whereas SEM it will scan the surface of the material. So, these transmitted electrons will give the inner structure of the samples. So, that is the beauty of the TEM. Now, the resolution is very high and the magnification is much higher compared to SEM, and yes, it can be used for the smaller particles. Now, you can see the dispersion, you can see the homogeneous dispersion, there is no noise in the formulation, you can see in the picture. So, and here also the AFM. So, AFM here we use a sharp tip to probe or interact the sample surface, generate the topographical map. Here you can get the 3D images of the particles. This data used to measure the height of the nanoparticles. Now, you can see it starts from 0 and it ends with the 117, which are same, and there is a black color, there is no size. And here you can see the same properties which we are comparing with the TEM. The resolution is high, magnification is very low compared with the TEM, and yes, it can be used for the smaller particle. Now, if you are more interested, you can go through this literature to know the basics and the principle of the SEM, TEM and AFM. Now, here we will see the principle. So, in the SEM, imaging is most commonly achieved by the monitoring the intensities of the either backscattered electrons or secondary electrons. In the picture, this is a sample, right? Your electron beam is passing, and these are the two types of electrons we will measure in the scanning electron microscopy, that is secondary electrons and the backscattered electrons. Whereas in TEM, what we will do? So, we will put the sample, the electron beam transmit, it will transmitted through the sample. So, here we will collect this and we will see the pictures. Now, we can see here there is an interesting study which done by one researcher. So, here they have prepared a mesoporous silica nanoparticles, and later they coated with the polymer. Now, they would like to know the surface or the coating of the mesoporous silica nanoparticles. Now, you can see the SEM A and B represents the capsules of mesoporous silica nanoparticles. So, whereas here you can see the same pictures in the C and D, which are performed by the TEM. Now, you can see the inner structures, the coating, you can see the layers. So, the inside is the silica and you can see the coating of the material. So, that is the greatness of the TEM. So, if you want to know the the details of your coating and if you want to see the inner structure of your material, so TEM is always preferable compared to SEM. And these are some other few characterization techniques which can also helpful to see the physicochemical characterization of the materials. So, for example, if you want to know the mechanical properties of your material, you can use universal testing machines, and if you want to know the electronic optical properties, you can use UV visible spectrophotometry and the Raman spectroscopy. And if you would like to know the magnetic properties of iron oxide nanoparticle, so the VSM is the one of the known and regularly used instrument to measure the magnetic properties. Vibrating sample magnetometry and the thermal properties, yes, you can use DSC and the DTA. And if you want to know the surface area and porosity, for example, if you have prepared a mesoporous silica and if you want to see the surface area and the porosity, so the BET method is a mostly used technique, and the structural and chemical composition can be performed. If you want to know the crystals, go with the XRD, and if you want to know the composition of your material, you can perform by EDX and FTR, that will give the chemical composition. And if you are interested to know the electrochemical properties, you can use XPS, that is X-ray photoelectron spectroscopy. So, with this you can also measure other physicochemical characterization techniques. So, these are the two references, these are the open access and you can see and you can utilize for more information. So, now we are moving from the physicochemical to the in vitro. As I discussed, in vitro studies, we try to see some cases, we use the various techniques to see the drug release, and sometimes we see the drug permeation, and we will see the drug loading, and we do some stability studies and in vitro cellular studies. Now, to understand the encapsulation efficiency. So, how we do the encapsulation efficiency? Very simple, after preparation of your formulation, you need to collect your formulation and transfer into the centrifugation tubes and do the centrifugation. Your particle will settle down and you can take the supernatant and check it by the any type of the analytical technique, maybe with the UV or from HPLC method, you will know the what is the free amount of drug in the supernet. So, this is another technique. So, here these are the centrifugation tubes comes in the market, which are also called as NanoCep. So, what you need to do? You need to put your formulation here, right. So, you have to do the centrifugation. After centrifugation, your free drug in a liquid form that will come into the bottom of the centrifugation tube. So, it comes in the different molecular weights. So, your particle will settle on the tube itself. So, later you can remove this outside and you need to collect this and you can check the free amount of your active ingredient. So, once you done with this and you can do the calculations. So, by using the encapsulation efficiency, what we need to do? You need to know the weight of the drug in the nanoparticle. Consider you have taken 10 mg of your drug in your formulation. So, now after centrifugation, if you got 8 mg, so weight of the drug in the nanoparticles, weight of the feeding drug, that is a 10 mg into 100. So, that will give the encapsulation efficiency. Whereas, drug loading, similar study you can perform, but here what we have to do? You have to consider weight of the nanoparticle, including your drug and the polymer, other surfactant. You need to consider the total weight of the nanoparticle and in that weight of the drug in the nanoparticle. So, here these two are the different studies. Okay. So, here you will see, you will compare with the total amount of drug and here you will compare with the total amount of the nanoparticles. So, these two are very important for the in vitro studies. Now, we in vitro drug release, as we prepare for the, we do it for the tablets. So, at different pH and a different time interval, we will see the release time. So, the release come from the your formulations from the carrier, right. We can compare in this way, like time versus cumulative release. At different pH, drug will show and your polymer also will respond based on the pH. So, typically you need to see release of the drug at different pH as well as at different time intervals, that is called as in vitro drug release or in vitro dissolution study. And stability study, this need to perform as per the ICH guidelines. So, here you can see, so at different, yes, we do the stability study to know the right storage conditions. This we need to perform at different humidity and the temperature conditions as per the ICH guidelines. So, this is a simple study for your carriers. You can see, measure the size at different time intervals. So, here is the researcher, they used one formulation and this 2 and the 3 formulations. At these three different formulations, they have compared their stability for almost 90 days, 3 months. So, their formulation is stable, there is no change in the particle size. The another one is the in vitro cellular studies. Here we use the cells to see the effect on the cells, whether cytotoxicity or hemolysis. We will see the release of hemoglobin from the RBC and also we can see the cell uptake. Now, here this is research done during my PhD. So, here what we did, we took the chitosan and chitosan coated surface modified PLGA nanoparticles. Now, we have shown that there is a uptake in the cells. So, by using confocal microscopy, you can see the presence of the particles by using the cell uptake studies. So, here in the third one, the in vivo preclinical studies. So, here yes, we can perform the pharmacokinetic studies and the pharmacodynamic studies and the toxicity studies. Yes, preclinical information that can be utilized by the scientific community to conduct first in human studies. So, if you want to go for the clinical studies, this data is very, very important, as we know that. So, pharmacokinetics can be influenced by the nanosystems, that is nothing but absorption, distribution, metabolism and excretion can be modified by using the nanosystems. So, that is why you need to compare your PK studies, pharmacokinetic studies with your conventional formulations as well as with your drug and the nanoparticles, right. It will affect the drug absorption and the distribution pattern as well as the metabolism and definitely excretion. Pharmacodynamic, yes, it will speak about the efficacy, potency of your drug. Once you modified into the nanosystem, definitely their efficacy will be improved and also you are targeting into the your cells and organelles and other organs, and efficacy also can be improved. Again, as you perform for PK, you can select some pharmacodynamic models. By with that you can compare with your conventional formulations and the free drug and then your nanoparticles, and definitely you need to compare the efficacy of all these three. And then toxicity study, nanoparticle itself can create a toxicity. So, that you must study the nanotoxicity of your formulation as well as your carriers. So, nanoparticles can protect the drugs from adverse effects, nanoparticles themselves can also be toxic. So, you must be studied without drug and with drug to see the toxicity of your formulation as well as only carrier. So, these are the studies are recommended to see the toxicity effect. So, systemic toxicity, that is acute, subacute and the chronic toxicity, then reproductive toxicity, local toxicity, hypersensitive toxicity, genotoxicity and carcinoid toxicities are recommended for the in vivo toxicity studies. And once you done with the preclinical data, so these are the drug discovery development stages. So, here once you done with this preclinical data, so here is the regulatory body you need to get the approval from the regulatory body. So, here you need to process, you need to apply for the clinical trials, that is a INDA, investigational new drug application. With this data you need to apply to the regulatory body. Once after the review, they will go for the, they will give the permission for the phase 1, that is for the, we will use for the human trials. There we will see the safety, and the remaining phase 2 and phase 3, this will perform the safety and efficacy study on the patients. The overall it will take 18 to 20 years. So, once phase 3 is done, again you need to go for the NDA, that is new drug application. After the several years from the FDA regulatory body, they will review and they will release, they will approve for the marketing. So, after that, after the marketing, there is a one more stage, that is the phase 4 clinical study. So, here we will see the side effects and other drug-drug interaction, food interactions that we can be evaluated through the phase 4 clinical trials. So, with this, so we are going to stop today's lecture. So far we have discussed on the various evaluation techniques. So, first we have seen the physicochemical properties and evaluation of the nanosystems, then we have seen the in vitro studies, and after that we have seen the in vivo studies, and then fourth one in clinical studies. So, we will stop today's session here. Thank you so much.